Nucleic acid hybridizations. A presentation from the Zoology Department GDC wrapper. Key concepts. In nucleic acid hybridization, nucleic acid or oligonucleotide populations, probes, are used to identify related sequences in test samples containing complex, often poorly known nucleic acid populations. Nucleic acid hybridization relies on specificity of base pairing. Nucleic acids in both the probe and test sample populations are made single-stranded and mixed so that heteroduplexes. Heteroduplexes are detected by labeling one nucleic acid population in aqueous solution and allowing it to hybridize to an unlabeled nucleic acid population fixed to a solid support. The stability of a heteroduplex depends on the extent of base matching and is affected by parameters such as the length of the base paired segment, the temperature, and the ionic environment. Key concepts. Commonly used DNA and RNA probes are hundreds of nucleotides long. Short oligonucleotide probes, less than 20 nucleotides long, can be used to distinguish between targets that differ at single nucleotide positions. Nucleic acids are labeled by incorporating nucleotides containing radioisotopes or chemically modified groups that can be detected by a suitable assay. Hybridization assays involve binding the nucleic acid population containing the target to a solid surface and then exposing it to a solution of labeled probe. In microarray hybridization, thousands of unlabeled DNA or oligonucleotide probes are fixed on a solid surface in a high-density grid format and are used to screen complex sample populations of labeled DNA or RNA in solution. The principal applications of microarray hybridization are in gene expression profiling and analysis of DNA variation. Terms to be familiar with. Anneal. To allow hydrogen bonds to form between two single strands. Forms a double-stranded nucleic acid duplex. Anneal has the opposite meaning to that of denature. Denature. To separate the individual strands of a double-stranded DNA duplex by breaking the hydrogen bonds by heating, or by exposing the DNA to alkali or to a highly polar solvent such as urea or formamide. Denature has the opposite meaning to that of anneal. Antisense RNA. An RNA sequence that is complementary in sequence to a transcribed RNA. Base complementarity. The degree to which the sequences of two single-stranded nucleic acids can form a double-stranded duplex, A binds to T or U, C binds to G. DNA chip. Any very high-density grid array, microarray, of DNA clones or oligonucleotides that is used in a hybridization assay. Feature, of a DNA or oligonucleotide microarray. The large number of identical DNA or oligonucleotide molecules at any one position in the microarray. Terms to be familiar with. Heteroduplex. A double-stranded nucleic acid formed by base pairing between two single-stranded nucleic acids that do not originate from the same allele. A heteroduplex may not have 100% base matching and so is normally less stable than a homoduplex. Homoduplex. A double-stranded DNA formed when two single-stranded sequences originating from the same allele are allowed to re-anneal. A homoduplex has 100% base matching and so is normally more stable than a heteroduplex. Hybridization assay. An assay in which single-stranded nucleic acids or oligonucleotides, the probe, is allowed to anneal with target sequences and form heteroduplexes. In situ hybridization. A hybridization reaction in which a labeled probe is hybridized to nucleic acids so that their morphological location can be detected within fixed cells or chromosomes. Melting temperature, Tm, the temperature corresponding to the midpoint in the observed transition from double-stranded to single-stranded forms of nucleic acids. Formation of probe target heteroduplexes. Sample, heterogeneous mixture of nucleic acid fragments. Probe, known nucleic acid sequences or synthetic nucleotides. Both sample and probe are denatured, raising the temperature. Now, sample and probe are mixed. We get 1 reannealed sample Na, 2 probe target heteroduplexes, 3 reannealed probe. Identification of probe target heteroduplexes. Hybridization assays involve binding the test sample population to the surface of a solid support. The immobilized population is bound in such a way that different sequences are fixed to defined positions on the solid surface. Both test and sample are mixed. Now, hybridization takes place. Then it is washed. We get probe target heteroduplexes. Probe target mismatches during hybridization. A. Longer nucleic acid probes, more than 100 nucleotides long, will form stable heteroduplexes with target sequences that are similar but not identical to the probe. This allows cross-species analyses and identification of members of a gene family. B. Short oligonucleotide probes are less tolerant of sequence mismatches. By using a high-stringency hybridization, it is possible to select for perfectly matched duplexes only, and so distinguish between allelic sequences that differ by just a single nucleotide. Hence, single mismatches are tolerated by nucleic acid probes, whereas it renders oligonucleotide probe to be highly unstable. DNA concentration affects DNA reassociation. DNA reassociation speed depends upon the initial concentration of the DNA sample. At high concentrations of complementary DNA sequences, the single-stranded DNA molecules finds a complementary strand and form a duplex quickly. The kinetics of reassociation can be measured using the starting concentration, Co, of the specific DNA sequence in moles per liter and the reaction time, T, in seconds, COT. This COT value, COT, the graphical representation is called COT curve, also varies depending on the temperature of reassociation and the concentration of monovalent cations. As a result, it is usual to use fixed reference values, a reassociation temperature of 65 oC and a concentration of 0.3 mNaCl. When a homogeneous probe is hybridized to a heterogeneous nucleic acid population in the test sample, the concentration of any one target sequence may be very low, causing the rate of reassociation to be slow. DNA labeling by Nick translation. Pancreatic DNA cuts phosphodiester bonds, P, to generate a 3-hydroxyl and a 5-phosphate group. These NICs provide the substrates for the multi-subunit enzyme E. coli DNA polymerase I. It has two enzyme activities. A 5-3-exonuclease, EXO, attacks the exposed 5' terminus and removes nucleotides in the 5-3 direction, and a DNA polymerase, POL, adds new nucleotides to the exposed 3'O group, continuing in the 5-3 direction. At least one of the dinucleotide triphosphates, DNTPS, is labeled red circle on CTP in this example, and thus label is incorporated at each C in the newly synthesized strand. Kindly look out for nucleic acid hybridizations Part 2. A Zoology Department GDC Raper presentation.